Yeah, we have my understanding is we have a pretty busy schedule today. So today we have very fortunate to have Dr. Lin to join us. Dr. Lin is a associate professor and autopsy director at Department of Pathology, University of Washington at St. Louis. But before that, she had a long time stay in the West Coast, did a PhD residency, breast fellowship and molecular fellowship. My God, how many more, right? All at Stanford. Very beautiful place. Then she moved on to the Washington 2018 and go all the way from assistant professor, now associate professor, pretty soon maybe full professor. Now, her research interest in inflammatory cardiovascular disease, including sarcoidosis, myocarditis, and all this transplantation. Without further ado, Dr. Lin, please, she has a very interesting agenda today.
Thank you. This is my honor today to share with you, and let me try to share screen and make sure that I can successfully do that. Okay. Oh, still wrong one.
It's good, it's good.
Is it good?
Go to your display settings and flip where it says display settings.
Yep, hold on 1 second. Oh, the opposite. Here. How about this?
Yes.
Yeah, much
Perfect.
better.
Yeah. Thank you, everyone. This is will be a fun journal club. I do not have any disclosure. The reasons I say it's fun is that I would like to do a little bit different format compared with the previous few very successful SCVP journal club. And instead of doing 1 to 3 papers in the kind of like center around the same thing, I want to kind of like modeling what Pulmonary Pathology Society has been doing for a while to go through multiple clinical, pathological, and translational journals and to highlight various papers that I think will be relevant and be interest of our CVP members. So as you can imagine, it's a very long list and a lot of papers. So I cannot cover all of the papers, so it's kind of like at the discretions, and I would love to, get some feedback and from the audience, see whether this is the interest of yours. But I also provide the list and summaries that you can access on the website. Mark just put that link in the chat, so you can access the PDF files. I want to focus on articles that focusing on human data and with potential impact of our daily clinical practice, because every single month there are a lot of great basic or translational cardiovascular research that we cannot cover in the reasonable time. In today's journal club, because we're experimenting this different format, I decided to go back to just 1 month in October, and except for our own journal Cardiovascular Pathology, that will be September, October issue, and then to cover all these papers. So here is the link that you can access a summary of all the paper that we are going to discuss and some of the paper that we won't have time to discuss today, but I provide a summary and then a list of paper that was published in the previous month in October. So these are the journals. After consulting some leaderships in SCVP, these are the journal lists that I experiment on this month, and focusing on pathology journal, here's the list. And clinical journal, initially I was going to go further down to the list, but I quickly realized that there are actually way more papers that is relevant to cardiovascular pathologists than I initially anticipated. So the gray out one, actually, I did not review those journals. So please check those journals if you want to see what's the most updated journal. And also the same thing happened with the research and translational journal list. Initially, I was going to go down further to the list, but realized that even just circulation research provide ample amount of manuscripts for us to discuss or to consider. So I tentatively put the manuscript I found in last month's publication into these categories. And then I want to focus on our discussion today in the first category, which is original articles, that I chose 8 papers ranging from somewhat translational to clinical studies to dive into a little bit. However, this won't be as deep and as comprehensive as what we have had for the past few journal club for obvious reason, because we have 8 papers and then we only have limited time. And then if the time allows, I also want to go through several more papers that I have reviewed and provide a summary that I think it's relevant to our cardiopathology practice, but more air on the clinical side for, important clinical trials in cardiovascular medicines. I also put some paper there that I encountered during my search, but I only reviewed the abstract, so I did not provide a summary of the finding of those papers, but people can quickly read through the title and see whether this is a paper of your own interest. I also listed a paper that we won't have a chance to discuss today for review consensus statement paper, as well as case report that I encountered during my search. So let's start with the original article for discussions. The first article is, first few articles from our own journal, Cardiovascular Pathology. The first paper I want to discuss is this novel insight into bicuspid aortic valve aortopathy. Long non-coding RNA, TOG1 and MIAT or MIAT are differentially expressed in BAV ascending aortas. So if you open the PDF file that I provided on the website, you will see that for each paper I provide an even more kind of like short version of summary or abstracts. But I also showed the original abstract from the paper for your reference. But basically, this paper is to investigate the possible underlying pathogenesis for ascending aortic aneurysm in patients with bicuspid aortic valve. What they did is that they include 29 patients that with bicuspid aortic valve and compare with 7 normal control, and then examine 28 possible long non-coding RNA by an RT-qPCR platform. And then they found that 2 specific long non-coding RNA is associated with bicuspid aortic valve aortic aneurysms. So here is some summary of the data from this paper. You can see that this is the volcano plot that 2 specific long non-coding RNA stood out from the rest of the 26 long non-coding RNAs. So they found that the TOG1 long non-coding RNA is downregulated in patients with bicuspid aortic valve, and MIAT1 is upregulated. And here's the relative expressions in the 2 cohort that is included in the studies. In general, they also noted that the patients, here you can see that the demographic is a little bit different between their experimental group and control group, that their patient with BAV is a little bit older. So they did, additional analysis and show that the differential express of TOG1 and MIAT is not due to the age bias between these 2 groups. The paper also presented an interesting study or analysis, but unfortunately, I do think it's not powered in this current cohort, is that they compare within the BAV groups that whether the patients has clinical significant aortic stenosis or aortic regurgitation or have a normal physiological aortic valve despite it's being bicuspid. And then they showed no difference between these 2 long non-coding RNA genes expression. However, I do think, the fact that it's not not statistic significance might be affected by the fact that the cohort is not powered to do such analysis. So that's the first paper. And then the second paper I included here again from our journal Cardiovascular Pathology is the cardiac macrophages density of COVID-19 infection relationship to myocyte necrosis in acute lung injury. So as a lot of audience here that we know, COVID-19 patient can have cardio involvement and with very significant morbidity and mortalities. And ample amount of literatures, also some from our SCVP members, has shown that the patient with COVID-19 infection can have increased macrophages in infiltrations in the myocardium. So this paper is diving into this topic in my mind, a very interesting angle is to compare 2 cohort, COVID-19 positive patients autopsy finding versus decedents have severe bronchopneumonia or diffuse alveolar damage, basically have a very significant pulmonary comorbidities or even cause of death, and see how the lung pathology can affect the possible strand on the cardiac pathology. So what they did is that they examined 10 COVID-19 positive autopsy cases and 20 non-COVID-19 but with severe bronchopneumonia or DAD decedents, and then to examine the prevalence of single myocyte necrosis and how the macrophages are distributed in epicardium and myocardium. So in their cohort, they did not see any definite lymphocytic myocarditis in either cohort. And they do found that 50% of the COVID-19 positive autopsy myocardium contains single myocyte necrosis. And when they examined the density of macrophages, they found that the density of macrophages is highest in COVID-19 patient or decedents with necrosis, followed by COVID-19 positive without myocyte necrosis, and then the control. So here are some of the highlights of the data from this paper. Here they actually show high power H&E pictures, how they define single myocyte necrosis. And as we all know, it can be very subtle. In their autopsy cohort, they examined 3 sections for each autopsy cases from right ventricle, left ventricle, and septate. And they group all up together, even if just one focus of myocyte necrosis was found, that case is being coded as presence. Here are their data of the macrophages enumerated by the CD68 staining in both epicardium and myocardium. And here you can see that on the upper panel are the epicardium compared with COVID positive versus COVID negative but bronchopneumonia patients. And here's the myocardium. And then lower panel is compared within the COVID positive group that patients with single cardiomyocyte necrosis also have higher macrophages account compared with COVID positive but without cardiomyocyte necrosis in both epicardium and myocardium, although the myocardium data is not statistically significant. These macrophages count are scored in hotspot under high power. And as you can see that there's actually quite a degree of variations of the macrophages count, especially when you compare between the COVID positive 19 and or COVID-19 COVID-19 positive versus COVID-19 negative cases. So it will be very interesting to see whether this kind of like cases that with higher macrophages count are actually in patients with different severity or longer or different phase of bronchopneumonia that might affect the cardiac pathology and physiology. The third paper I included again from our journal is the histopathological evaluation of chronic rheumatic mitral valve stenosis, the Association with Clinical Presentation, Pathogenesis, and Management at National Cardiac Institute, Tanzania. And I found this paper quite interesting and very educational to read because, to be quite honest with the audience, that in our daily practice nowadays, I very rarely come across the rheumatic mitral valve anymore in my current practice, although we all learned that from medical school and residency. So basically, this is a prospective study of 54 rheumatic stenotic mitral valve in this center. They actually collect this cohort within 1 year and then did the analysis by routine H&E scan, von Casa scan for calcium, and IHE scan to evaluate the inflammatory infiltrate. And they found that consistent with the known literatures that high calcium deposit can be found in patients with older age. Their definition is older than 30 years old, male, and with severe mitral valve stenosis, although that's just a trend, it's not statistically significant. And the inflammatory infiltrate is also associated with the degree of calcifications. So here are some beautiful pictures showing the pathognomonic findings of the rheumatic mitral valve stenosis, including the apologize if I didn't say this correctly. The Ashkov bodies and the interstitial cells in the valve and showing the increased inflammatory infiltrate and neovascularization. Here on the HN stain, you can appreciate very significant calcifications, and that can be shown on the von Casa stain again. And here are just some representative pictures of the IHC stain of the inflammatory cells. So here is the chart that is in this paper to show the correlations or between calcification and some clinical variable. As I mentioned, that although there's a trend that older men with severe stenosis have higher calcification, it's not statistically significant. The next paper, by the way, I think there's some note from the chat. I am not sure what's the best way since we're experimenting this. Should we stop discussing this paper, or Peter, what's your thought?
Yeah. So Mark, do you have any comment, or Jeff?
Jeff and I think we're maybe having a public discussion about the paper in the chat, which I don't know. We can keep some stuff going in the chat to comment on what you're talking about rather than stopping you, if that's okay.
Yeah.
But I think there was a specific question to you from Henry Tazalar, though, for the first paper. Does it seem like the right control group for that study? So the original long non-coding RNA study you did, if you saw that.
Flip through.
Yeah, they had normal controls, I mean, assuming patients without aneurysms.
Yeah, I agree.
Why not pick people with aneurysms who had tricuspid valves if they're really trying to delve into the difference with bicuspid valves? So
I agree with you, Henry. And then also I think that's also, although they try their best to have multiple way to try to mitigate the difference between the age group, but we all know that the aorta will go through degenerative change even just in normal general populations. So I agree that the control group is not ideal in this particular setting. Okay, I'm just going to keep all through the rest of the paper, and then
Yeah, please.
the next one is from JHLT, is the circulating microRNA in cellular antibody-mediated heart transplant rejections. This seems to be a recurrent thing in the recent literature to try to identify a non-invasive method to perform cardiac allograft surveillance for both acute cell rejection and antibody-mediated rejection. So in this multi-century cohort, basically they've identified a small subset of microRNA that can discriminate no rejection versus acute cell rejection, or no rejection versus antibody-mediated rejections. Then they validate their finding in the validation set and an independent Stanford cohort. So here based on their data, based on those findings, they actually create a score. There's some echo. Hello. They create what they call ACR score, AMR score, and show that in their control pre-rejection, rejection, and post-rejection, there's a uptrending from control to pre-rejection, and then kind of downtrending after the patient recover from rejections. And then I think the finding is very encouraging, but I do believe that this score, although it's statistically significant, you can see that they have a wide range overlap. And then with the numeric difference is also somewhat limited. So how this can be practically used in the clinical setting, I do believe that it requires further studies or additional cohort to examine this proposed ACR and AMR score. Here, I also want to just put table 3, which is that the comparison between pathologist review and then the original pathologist review. As we know, and actually discussing the first SCVP journal club, there are inter-observer variations in both ACR and AMR grading. The next case is is a brief communication from JHLT from the Duke group to report the institutional experience of the first 14 thoracic transplant using COVID-19 positive donors. And then what they shown in this early cohort is that there's no unexpected acute cellular rejections with pretty good patient survivals. There's no recipient seems to develop COVID-19 symptom or being, quote unquote infected due to the transplant from a COVID-19 positive donors. They do have 2 adverse outcomes both for heart transplant. One patient actually required retransplant within 1 week due to the hemorrhage and hypercoagulability. I'm going to show you the gross and microscopic finding. And the other patient unfortunately died due to aortic anastomosis breakdown and then infections. So here is the gross examinations of the retransplant heart for patient number 4 in their cohort. They also noted that they found the endomyocardial biopsy from this explant, is positive for nucleocapsid immunohistochemistry stain. However, when they perform PCR studies using the myocardium tissue, it's actually negative. So the significance of such staining is still unknown. The next paper is also from JHLT, and then this is specifically interrogating the different typing methodology in the pediatric heart transplantation group and see whether we can improve patients' outcome and decrease the possible donor-specific DSA formation after the transplant. So in this cohort, paper, it's a single center studies. They retrospectively analyzing the 77 pediatric patients underwent heart transplant. Each patient have the traditional HLA typing, and this is what they call an epilet mismatch analysis. And then they found that a specific HLA class 2 DPB epilet mismatch seems to associate with graft loss, but not other type of epilet or the traditional HLA antigen mismatch. So I need to admit that despite my best effort to read this paper back and forth, as an AP trend pathologist, I have my limitations on HLA and then this methodologies. So I actually kind of like copy paste like the method section here for people of interest to show that's what's the novel method they're trying to compare in this particular manuscript. But basically, as a multivariate analysis, using either epilet mismatch or HLA allele mismatch, there are some trend or even statistically significant changes compared with the outcome, which is graft loss, AMR, or ACR. However, when they did the multivariate analysis, as I mentioned in the previous PowerPoint, only the HLA class 2 DPB epilet seems to be associated with graft loss. So I think this is an interesting paper to try to conquer the kind of like a very challenging area in heart transplant, which is pre-sensitized patients, especially in pediatric patients that have multiple transfusions, and how can we better handle patient clinically and prevent graft loss. The next one in this category is research later from circulations. So basically what they did, it's defining the fetal gene program as single cell resolution in pediatric dilated cardiomyopathy. So the background of this such study I found is quite interesting is that it's been known for a while that there's a kind of like a dogma in the cardiovascular research that when postnatal heart gets stressed, they can turn on or like switch back to a kind of like a fetal gene expression. The best, most known example is like switching between the alpha myosin heavy chain and the beta myosin heavy chain. So this group, they used human sample and did single nuclear RNA sequence using fetal heart, non-disease heart, and heart from dilated cardiomyopathy, and then to examine the transcriptomic profiles in different constituents in the human heart. And what they found is that indeed there's evidence of reactivation of fetal genes found specifically in cardiomyocyte and fibroblasts. And it's not all the fetal genes have been reactivated. It's about 8% of the fetal gene has been shown can be reactivated in the dilated cardiomyopathy. So this is a very busy chart. I'm not going to go through the detail, but basically these are the genes that CM is cardiomyocyte, FIB is fibroblast, that they found it's like specifically upregulated or downregulated in cardiomyocyte and fibroblast that is being switch back to the fetal phenotypes in this small cohort. I found this is a very interesting preliminary data, although I would love to see more follow-up data with validations of the setting. This paper only presented the single nuclei sequencing data and in a relatively small cohort. I believe this is the last paper for the more kind of like discussions for today is the genetic architectures of acute myocarditis and the overlap with inherited cardiomyopathy. So as we know that acute myocarditis might trigger subsequent dilated cardiomyopathy or arrhythmogenic cardiomyopathy in patients. So the authors they examine this hypothesis that see whether we will see DCM or ACM related genetic variants in myocarditis patient and how they affect the outcome. So this is from two cohorts, one from England, one from Netherlands. They have 333 consecutive myocarditis patients, and they sequence these genes that has been known to link to either DCM or ACM and compare with large healthy cohort controls. And they found that in these almost at 400 myocarditis patients, 8% of the patients actually do have a pathogenic variant that in these, 16 genes that is known to associate with DCM or ACM, compared with the control is less than 1%. That's the healthy control exhibit any pathogenic variants in these 16 genes. They also found that it could be clinically significant because the 5-year mortality risk is higher within the myocarditis patient if they do have a genotype positive, which means that exhibit at least one pathogenic variant in these 16 genes. It's not it's not statistically significant, but the numeric difference is 3% versus 11%. And then they found that depends on which cohort they interrogate, the England cohort, the Netherlands cohort, it seems that the DSP and TTN truncations are highly enriched in these acute myocarditis patients. So here are some of the data from the, from this paper. You can see that in the London cohort and in the Netherlands cohort have a little bit slightly different genotypes, even though they are interrogating the same type of disease, the acute myocarditis. And here are different, different type of genotypes that is associated, seems to be associated with different levels of LV ejection fraction, which kind of like a surrogate of the clinical outcome of the heart failure. And it seems that the DCM gene, that the pathogenic variant DCM gene patients seem to have lower LVEF in their cohort. So I very quickly use like 20 minutes to go through 8 papers. To be honest with you, as I say, thank you all for this trial, because like this is like a new format, and then I'm going to go through the chart, the chat, and see whether we want to discuss some of the paper before we go into the next part of this journal club, which, you know, another 7 paper that I think could be clinically relevant, but may not require as much discussion as the 8 paper I just presented. We already did this. Mark, do you want to?
Well, I think Jeff Sappitz brings up an interesting point about that last paper. Maybe it's worth discussing that some. I see Melanie chimed in as well. Jeff, do you want to discuss? I think that's really interesting.
Yeah, sure. I mean, so maybe I'll just quickly tell you a story about the first time I ever diagnosed arrhythmogenic cardiomyopathy when I was at Washington University. It involved a young woman who kept on having appropriate shocks from an ICD that had been implanted. They didn't really know what was wrong with her, except she kept on having, you know, spontaneous ventricular tachyarrhythmias. she was getting shocked so frequently that she decided she couldn't live this way anymore and requested that something be done. And so she was given a heart transplant. And the heart came to me and I diagnosed arrhythmogenic cardiomyopathy. It was a rather clear-cut case. I later learned that her father had died a few years earlier at age 43. This was before we knew anything about the genetics. He died at home. He was autopsied by a medical examiner in Illinois. The heart went to the Mayo Clinic to be reviewed, and the diagnosis was myocarditis. The problem was that what they sent to the Mayo Clinic was only pieces of the left ventricle, slides of the left ventricle, not from the right ventricle. So I was able to retrieve the entire case, and the RV showed clear evidence of arrhythmogenic cardiomyopathy. The point is simply that inflammation can be very prominent in arrhythmogenic cardiomyopathy. It can occur in the left ventricle. It can occur in areas that don't show a lot of fat and other characteristic features, and it can easily be mistaken for myocarditis. Rather, it would be called myocarditis implicating a virus or something, rather than a familial cardiomyopathy. So in this paper that Che Yu just presented, which I did read, but I'm trying to remember, I recall, did they really have tissue diagnosis? How did they make the diagnosis of myocarditis?
Well,
And-
they use tissue diagnosis, also cardiac MR. And then
Yeah.
they say they use the little criteria with both blind review and central review.
Yeah, I just think it is possible, maybe even likely, that they misdiagnosed some cases as myocarditis when they were actually familial cardiomyopathies known to have a very prominent inflammatory component. And the desmoplakin variants in particular, which typically give left-sided disease, very hot findings with imaging. When you look for inflammatory markers by various imaging studies, the desmoplakin patients light up. So I just think, first of all, it's an interesting paper. I think it makes a good point, but they may have just made the wrong diagnosis in the beginning, quite frankly.
Yeah, I totally agree. And I think Melanie also just pointed out especially for the DSP mutations. There's one common, say, artifact by Stephen, but I'm not sure which paper that was about.
If I can jump in though, to
Yes.
what Jeff is saying, I remember like a decade ago, Christina Basso had a case of like a 8-year-old boy that looked all the world like myocarditis histologically, but then had the diagnosis, they did the genetics, and it was arrhythmogenic cardiomyopathy. And I've always wondered from then if whether a virus or something could be triggering. Jeff, I'd like your thoughts, whether there could be some environmental trigger that some people just, it goes away, but because they are susceptible, then they proceed down this road. As you know better than I do, a lot of patients who have mutations are clinically fine, and maybe they haven't had the trigger.
Yeah, it's a great question, Steve. And I think there's a lot to what you say, although I don't think there's any definitive information to say one way or the other. You're quite right that the penetrance is quite variable. It's very common to have families with a proband who may have died suddenly, and then you identify other members of the family who have the same allele, but who don't seem to have the phenotype. So that's quite variable. And this idea of triggers, I think, is very likely. Probably the most important trigger is exercise in many patients, because that's clearly known to increase penetrance and increase risk of adverse events. But a viral infection might very likely be an important trigger, which in a person who didn't have the risk allele might have produced little, if anything at all, but in somebody at risk might produce a much more impressive clinical picture. So I think this is sort of where we are in understanding the role of inflammation, and whether it's due to viruses per se, or whether it's a combination of triggers and risk alleles. I suspect it's all of the above, and we're only beginning to figure this out.
This is actually a great discussion. It's kind of a great segue to the first paper into the next part of this journal club. This is another paper from our journal Cardiovascular Pathology, examining the Coxsackie virus and adenovirus receptor expressions in both cardiomyocyte and the rule platelet in patients with dilated cardiomyopathy. And then, so this has been known, especially in animal models, that this specific receptor may play a role in the pathogenesis of inflammatory dilated cardiomyopathy. but there's limited data how this specific protein is expressed in human in patients that with myocarditis and dilated cardiomyopathy. So they examined the endomyocardial biopsy and actually electron microscopy of the platelet, and then the serum level of the inflammatory signatures in patients with inflammatory dilated cardiomyopathy, and then compare with the a healthy control cohort. And they found that upregulations of the CAR, this virus receptor is found in higher in both the endomyocardial biopsy and the platelet from the DCM compared with the control cohort, and also associated with higher inflammatory signatures in the serums. So for this, the next 7 papers, I didn't include any of the figures or tables, but people who are interested is, the references in both the PDF files and here in the PowerPoint. So the next few papers, there's a hand from Henry.
Yeah, thanks. So, maybe I recognize I may have been living under a rock, but can somebody tell me about inflammatory dilated cardiomyopathy? I don't know that as a accepted terminology.
I don't think I used that terminology as well, but it was used by this particular group. I will welcome any kind of comments from the group.
Well, so if I might comment briefly on that, because I've been trying to get my arms around this as well. So I think we all recognize that there's some patients who carry the diagnosis of dilated cardiomyopathy, who seem to have a more active arrhythmia kind of picture than others who have more of a heart failure type of clinical picture. And clearly there are some patients with dilated cardiomyopathy who die suddenly. It's now being shown that certain variants in certain DCM genes might be linked to a more arrhythmogenic phenotype. So for example, lamin A is one of the ones that most likely causes that phenotype. Filament C is another. There are some phospholamban and some Desmond variants that have also been implicated in what is now being called arrhythmogenic DCM, although I'm not aware of any hard and fast, kind of criteria that you use to decide if it's DCM, if it's a DCM or regular old DCM. But in any event, I think it again reflects just the diversity of the clinical phenotypes. The genetic era has ushered in a level of complexity that far exceeds our ability to categorize disease. So calling things, HCM, DCM, and ACM is probably not going to be terribly useful. Maybe calling things myocarditis may not be terribly useful in the future as well. So anyway, that's what I know about arrhythmogenic DCM.
The next few papers I kind of purposefully select, to highlight some of the recent important clinical trial in cardiovascular medicine. Oh, there's a chat again.
Thank you.
Oh, okay. There's a series of several important trials that has been published recently, may or may not be immediately applicable to cardiopathology, but I always found that the clinical relation and understanding what our clinical colleague is thinking is very interesting and important. So again, we'll see whether this is something SCVP member is of interest. One of the studies is the protected trial, but basically is that for TAVR, several centers and actually some of the local centers also use this type of kind of filter to capture the debris that is generated during the procedure to prevent cerebral embolic event. However, this is actually the first large prospective studies, multi-center, actually multi-country, to examine the efficacy of using such device. And surprisingly is that they show there's no difference for their outcome with or without using this device to capture the debris that is generated during the procedures. Another trials, the revived, is another kind of a long-standing question in intervention cardiology, is that whether there's any benefit to perform PCI in patient with ischemic cardiomyopathy that basically is not acute chest pain, it's not a acute myocardial infarctions, but it's kind of like already into the remodeling phase. Is there any benefits to perform the revascularizations in these patients? Again, this has been variable in practice based on the centers and the anecdotal experience. And in this large cohorts, they showed that there's actually no difference in their primary outcome, which is death or hospitalizations, or even secondary outcomes compared with or without PCI in this particular patient group. Another trial I want to highlight here is actually a population-based screening. So this is a trial that's in publishing NEJM that with almost like 50,000 men in between the age of 65 to 75 in to enrolled into screening versus not. When they say screening, cardiovascular screening, it's actually a non-contrast CT to see how much calcification is in the coronary in the coronary arteries, blood pressure measurement, and some blood test. And the primary outcome is as a death with 5 to 6 year median follow-up. And actually they shows that, again, personally, I think it's a surprising finding is that with this kind of like screening that intuitively we will think it will be beneficial to capture some early event or patients with severe CV risk actually did not have cause different outcomes between the screening and the no screening group. This is another trial that is recently published in Lancet, is the TIME study. So this is also another kind of debate in clinical cardiovascular medicine for a while. That's where they're taking antihypertensive drug during the evening is more beneficial than in the morning, because then one of the rationale is that it's actually protect through the night, and then a lot of like hypertensive spikes actually happen in the early, like midnight or early mornings. This is another large cohort that is randomized into morning versus evening dosing group. And this is another trial. There's no difference despite the common sense or belief that evening medication is a superior regimen. Last but not least, clinical trial to highlight recently that is catching attention is the OHeart, is to use allopurinol to treat a patient with ischemic cardiomyopathy. So they enrolled almost 6,000 patients that without gout, because as we know, allopurinol is a medication for gout, and there's no difference in primary outcome for ICM patient treated with or without allopurinol. And again, I purposefully kind of like flip through these papers in a very fast speed. I mean, as you know, all this kind of like a large multicentric clinical trial study comes with a lot of nuances, like what is the patient population, what is in the intention to treat, with a lot of things. But I found this like kind of high levels, you know, knowing what's going on in the cardiovascular medicine. Hopefully it's like something that will interest our group of audience. This is actually an interesting paper. Is they report 147 individuals with MYH7 variant may or may not have had clinical manifest of dilated cardiomyopathy, and then track this patient and see what's the natural history and clinical pathological correlations for patient that carrying pathogenic variant of MYH7 genes. So they found that with a median follow-up of 5 years, they found that most of the patients actually already developed dilated cardiomyopathy when they enroll the patients. And then even if they are initially considered negative in the enrollment period, 1/4 of them actually develop DCM within that 4 to 5 years follow-up time. So they found that the MYH-related DCM is actually characterized by early age of onset with high phenotypic expressions, which means that most of the patients actually develop DCM either before or after the enrollment. And then with frequent progression to end-stage heart failure, with some of the patients actually require heart transplantation. Here are just some of the data from this manuscript and showing different type of pathogenic mutations in different segment of the MYH gene, which is a large gene, and they may have somewhat different clinical manifestations and outcome. So again, this paper have a lot of like very comprehensive analysis, but I just want to high level mentioning that this have a paper that's diving into how the genotype translate to phenotype in the large cohort. There's no chat. The following 7 paper, I need to admit, these are the paper I came across when I review all the journals I listed previously, and I did not provide any summary in the PDF or here because I only read the abstract, so I do not feel comfortable to provide a kind of like my understanding or in the nutshell like what these papers are about. But I hope that this will provide kind of like a reference for people that if you find like a specific topic that you have interest in, these have been published in the past a month or so. And there are also like 5 review paper, consensus paper, statement paper that I want to highlight here, especially the first one that's from Dr. Basso, a very, very comprehensive clinical pathological review of myocarditis published in NEJM. You know, it was very intense, but very educational reading, at least for me. And then I found that is a very up-to-date review for myocarditis. And there are several others that's kind of like peripherally related to our specialties. I also want to highlight the case report I encountered during the search. And again, you can kind of like somewhat pick up what type of case report they are based on the title, except for this one, which is the NEJM, one of those like case presentation. It's actually eosinophilic granulomatosis with polyangiitis that's affecting the epicardial arteries. So thank you everyone to endure this different type of journal club. I need to say that, you know, I ran this by the leadership and say, hey, you know, we have a successful run of, you know, several journal club by now. Do we want to try a different format? And I need to say that this is not what I thought it would be when I proposed this. I found this from my, I would love to hear, and I believe like, you know, Mark and other leadership would love to hear from your opinion and feedback that, you know, whether this type of format is useful. From my personal perspective, when I prepared this, here are some of my thoughts. First and foremost, I did not realize how many papers there are each month that is relevant to cardiovascular pathologists until I start to tally it. It's actually way more than I thought, and it's actually difficult to pick. I hope that the PDF file that I provided online is comprehensive. I try my best, but even, you know, I apologize if I miss paper, especially if published from your group. The other thoughts I have is that to be from my perspective is that I think I swing to another extreme is that I feel more like a curator to listen to all the paper rather than be able to dive into constructive discussions or constructive critique of highlighted several papers. So, and this is also like takes way longer than I anticipated. I initially thought that going through this 22 journals should be quite fast. Just to give people or you know, future presenter kind of like an idea. If you haven't done PPS before, I personally haven't done PPS before. I have no idea like how extensive this could be. To prepare this journal file, it took me more than 12 hours to just go through paper and read paper. As I said, I read about like, you know, 15 to 20 paper in all of this. The paper I didn't read, I didn't provide a summary, and it's a very extensive kind of like overview. And so this is what I have and what I presented based on the screening of the journals I listed that's published last month. So I would really appreciate any feedback about, of course, the articles first, and then also maybe some feedback about whether this is a useful format for the audience. I'm going to stop sharing screen.
Thank you so much. This is very covers a lot. Obviously, you spend a lot of time, so it's interesting, and I think we learned a lot too.
I guess
I'll chime in just really quick. This is, you know, first of all, I'm sorry I'm a little bit late. I had clinical service until just about 20 minutes ago. But I also enjoyed at least the element of the format that I saw. I think it was pretty analogous to what we have done over on the pulmonary pathology journal side for a while. And in terms of the time commitment to preparing it, it seems to be roughly on par with that as well. I see both Brandon and Henry are here, and they can attest to that fact. Certainly, it's not uncommon to spend 10, 20 hours in preparation for one of those journal clubs. Fortunately, you only have to do it once a year, unless you get a year off, which is sometimes kind of nice, right, Henry? But
I'm counting on
yeah, it's a lot of work.
2023 for me. Yeah.
Yeah,
Another round of
2023 is my first year off, I think, in 14 years. So I'm ecstatic.
I think it's great. Do people have comments though, going back to these wonderful presentations of these papers? Is there any paper that really is bugging somebody they want to discuss or come back to? No.
I think just the ongoing issue with myocarditis. I'm eager to read Christina's article. And I think the beauty of this and the way we run the Pulmonary Journal Club, which Jeff Myers and I have been doing for, 33 years or something, is that, you can't cover everything that everybody's interested in, but we pick the number of journals. We outline the journals that we're going to review. So, I think if you move to this format, it'd be nice to identify the 15 or 17 journals you're going to review, and then pick out the articles that, you think the group is most interested in. But then everybody else can look at the other stuff that you just list briefly, JU did this time, just to list the stuff that she didn't have time to go over. And then you can go back and read those, but at least you don't have to look at all those journals.
Oh, I love what has been put in the chat by Stephen, that I think, we should have probably, consider a hybrid kind of formats that go, because I would love to dive deeper into some of the paper to talk about the results, the limitations and possible future directions, and elicit more organic and interactive, discussions. But that's why we call it the experiment.
You did a wonderful job summarizing all of these. Thank you for taking the time to really work through all of these and provide this documentation for us and this wonderful journal club. Kind of listening to the feedback from Joe and from Henry and from you with regard to the time it takes, I wonder if we should leave it up to the presenter and let it kind of be still sort of an organic thing. Do you want to do a more exhaustive kind of summary, or do you want to focus more on an in-depth? I don't know that we need to have a prescriptive type of approach. We could simply let it happen and allow the presenter to really choose what they bring to the table.
Oh, just to kind of another thoughts that kind of like crossed my mind when I was preparing this. I noticed actually cardiovascular pathology is extremely underrepresented represented in most of the PATH journal, because as you can see, I went through all the PATH journals, but I only actually found one paper, I think in the Journal of Pathology. That's it. There's, in most of the journals, we don't see any cardiovascular. And I'm not saying that we should publish otherwhere else than our home, cardiovascular pathology journal, but I think from the perspective of like recruiting young blood to our specialties, like, a lot of our residents, they read like MALPATH, AJCP, AJSP, they don't necessarily will pick like cardiovascular pathology. The other things I notice when I go through the journal is I know, several members in our society have strong forensic or autopsy expertise and interest, and those articles are also underrepresented in the current roster. That's just like my 2 cents. Yeah. All right. Mark and Peter, I guess we can call it a day, and then everyone can enjoy 5 more minutes of the our busy schedule.
Yeah, we can give back 5 minutes, right, to everybody? Mark?
Yeah, that sounds great. Happy Thanksgiving.
Happy Thanksgiving, everyone. Thank you. Thank you.
Thank you all.
Great job.